Hi everyone, I'm Jennifer. Hi everyone, today I'm gonna talk about a powerful imaging technique using by the confocal microscopy. And this imaging tech was used by a zero lab at UNC, and they are using this imaging tech to see how the liver cells recover from damage. So we can start with a very big question. How do scientists see what happens inside an organ? And for the background information, the liver is one of the only organs in the body that can grow back after a damage. However, all the repairing actions are appears on cellular level deep inside the tissues, which we cannot directly see them by our eyes. And those repairing actions including cell dividing, cell moving, and cell rebuilding. So in this way, we really need a powerful tool to do the observation. And here comes the confocal microscopy, and it is a superpower camera that can take pictures inside an organ without breaking it up. And here's a picture of the confocal microscopy. It looks very complex, more complex than the regular microscopy we use in labs. However, the mechanism is kind of easy to understand. Imagine an organ as a loaf of bread, and in order to eat the bread, we have to cut them into thin slices. And after we cut it, we can still stuck them back to form a whole bread. So that's exactly how the confocal microscopy works. Firstly, they will scan and photographing slice of the organs, and then they will take pictures for each thin slice inside the tissues, and finally they will stuck all the image back to make a 3D image of the organ. So, as long as we can see the image, how to distinguish between the different cells? Because for example, we have liver cells, we have blood vessel cells, we have nerve cells. So here the scientists use different fluorescent labeling. They will highlight different cell types with different colors. For example, they will label the red blood cells as red, and they will label the blood vessel cells as blue, and label the nerve cells as green. So in this way, it will help scientists to see what each cell's type is and what each cell is doing for repairing the whole organ. So how do the Azio lab use this tech for? They use it to watch how the blood vessels reconnect after the vessels are break, and also track how the nerve cells are sending signals between cells to cells. And in this way, the liver cells work together and repair the damage. So why this technique, the imaging technique using the confocal microscopy is important? Firstly, the liver injury is not common among populations due to over intake of fatty acid and alcohol. And so in this way, studying regeneration using the confocal microscopy can help the scientists to develop some potential treatments to improve recovery of organ damage for liver, and also can for other organs such as the heart and like your stomach, for example. So here the link is very clear. If you have an organ injury, and then the scientists will observe that under the imaging technique using the confocal microscopy, and they will have a better understanding, and finally they can have a better treatments. They can come up with a better treatments. So here are some main takeaways. Firstly, the confocal microscopy gives scientists a clear window to observe regeneration. And secondly, it helps scientists to study regeneration deeper. And finally, it can offer a step closer to improving healing and developing new treatments. Thank you for your listening.